You got that. Okay.
Yeah. Hey, hello, everyone. I'm really excited to introduce to you in person Genevieve Mount, who has been a postdoc in my lab, but virtually because she's co-advised by Molly Womack at Utah State University. Genevieve did her undergraduate at UC Davis, where she happened to work with Chris Martin on pufferfish. She worked after that, worked in Brad Schaefer's lab. So applied for a job in Davis, got moved to UCLA, and helped Brad set up his next gen lab, which is pretty exciting. Then after that, she did her PhD at Louisiana State University with Jeremy Brown and Chris Austin. The title of her dissertation is Quantifying Structure and Variation in Complex Phylogenetic Data. She had a paper come out earlier this year in SysBio from her PhD. So if you're interested, the title is Comparing Likelihood Ratios to Understand Genome-Wide Variation in Phylogenetic Support. So if you're interested in uncertainty in phylogenetics, Jenna has offered kindly to make time tomorrow. I sent her on a spreadsheet. I believe there are still some times left. I'll also mention that Genevieve is our tweeter for AmphibiaWeb. So if you follow AmphibiaWeb on Twitter.
Oh, I definitely forgot to do that. Yes, on Monday.
Don't say that. No, but it's been really great. You know, it's awesome to have a social media presence. AmphibiaWeb really appreciates that. And I have a fun fact here that we dug up. That Genevieve had a short but distinguished career at Noah's Bagels, after which she promptly took the GRE.
Yes.
Okay, Genevieve, thank you.
Awesome. Thank you. And thanks for having me. I realize this is my first talk in like 3 years in person. So hang with me. It'll be great. And this is also kind of an evolution of my dissertation talk, trying to slowly morph it towards a job talk. So it'll be a little bit more conceptual, hopefully. So yeah, I'm going to be talking about phylogenetics and frogs today, talking about methods, morphology, and conservation. And so my PhD was mostly in phylogenetic methods, which I think are awesome, but are also really difficult. And the main thing that I was looking at is leveraging all the variation in this big next gen data that we've been making for a while now. And so that section of the talk will be featuring trees, genes, and morphology. And the second section is going to be frogs, which are just awesome, not difficult. And I'm going to be talking a little bit about evolution of trait loss, which is my current project, and conservation genetics. And I will not be featuring vocal sacs. So phylogenetics, I know probably everybody in this room is convinced that it's needed and interesting. And the goal generally, just to give an overview, is we're trying to reconstruct evolutionary history of animals, right? And so we can use that evolutionary history for a lot of cool things, like learning about evolution, like most people in this room do. But we can also use it for other things outside of evolutionary biology that we might not always be thinking about. It can be used in forensics, looking at origins of HIV transmission. It can be used in virology and infectious diseases. Now there's like a website where you can look at the phylogeny of COVID. And it can also be used in drug development to look at the evolution rates of different proteins and use that to target parts of the molecule for drugs and to attack. And so my pitch is that phylogenies save lives, and that's why you should care about this part of the presentation. And so with our goal to reconstruct evolutionary history, we have a lot of data going into this. We've got our animals that move on to, we extract genes or morphology, usually end up with a big set of trees that essentially we smush all this together into one reconstruction that we use for downstream analyses to test hypotheses and do other research. And so put very, very broadly, we're basically averaging across all of this information in the data to get our single tree. And the problem is that we're losing a lot of information by averaging across all that variation. And just to give you a very obvious example, the average of both of those distributions is going to be the same, but they have very different signals, very different information. And so what I'm really interested in my research is whether or not our single tree is missing information that's relevant to our questions. So like I said, big data has lots of variation, and there can be conflicting signal. And that's basically, when I talk about signal here, I'm defining it as just information in the data that supports one topology over another, one reconstruction over another in terms of who's related to who. And so I'm interested in looking at, is there support for different topologies in our data? And the sources of this can be biological and methodological, things like convergent evolution, horizontal gene transfer, misidentification in your data, or inappropriate models. So what happens if we miss the information? You end up with inaccurate inferences, inaccurate historical relationships, evolutionary processes, and you're missing information about how molecular evolution is happening. And so I'm really interested in extracting these larger patterns of variation and learning more than just like, okay, here's our average tree, but like, okay, what's the distribution of trees look like? And what are ways that we can quantify that in a way that's helpful and meaningful and isn't just staring at like 100 trees on a page, because that's not actually useful. And so like Becca mentioned, my dissertation was basically identifying and quantifying variation. And so asking, does our data support multiple topologies, and what kind of cool information can we find from that? And all of the approaches that I've worked on so far are pretty agnostic approaches, meaning there's no assumptions as to where the conflict is coming from or what's causing it, or the evolutionary processes that are creating that conflict. And I have leaned towards these approaches because my background has been in herpetology, and working on just developing next-gen lab work for amphibians was difficult because they have giant genomes. And so I was finding a lot of resources wouldn't apply to things where you didn't know what the genome was a priori, or you didn't know what was going on in the system. Because there are definitely more methods out there that look at conflict, but are assuming incomplete lineage sorting or horizontal gene transfer. And so I was really interested in methods where, okay, we don't know anything about this system, but we want to know if there's conflict in the data. And so I'm going to go over 3 different kind of levels that I've looked at this at. First, I'm going to go over trees, so identifying broad patterns in like a set of trees. Then I'm going to look at genes and looking at, once you already know maybe what the potentially 2 major conflicting topologies are, what are the support, and then also comparing some methods that you can use to do that. And then third, I never would have guessed that I would have a project working on morphology, but I actually ended up also looking into morphology and how we're quantifying how characters support different topologies. So first, I want to look at, can we identify some of this broad variation in our huge sets of trees that we're kind of smushing together to get this one hypothesis of evolutionary history? And so sets of trees can come from a lot of different places. It can be a set of gene trees where each of your loci is a different tree. It could be that you're comparing different analyses. It could be, or different pipelines of how you've done your analyses. Or it could be looking at a posterior distribution. So maybe you're trying to diagnose MCMC mixing issues, stuff like that. And so essentially what I want to do is group trees that contain similar information. And so what's the point? Why do we care, right? Is we're trying to find what that conflicting signal is. If you don't know already, okay, yeah, there's incomplete lineage sorting, or I think that there's this conflict in my data. If you just have all the trees, you're like, okay, well, is it just one simple average that all supports the same thing? What are the major signals? And so you can use this to find conflicting signal, outlier loci, like I said, posterior distributions, or evaluate mixing. And so essentially what we want to do is go from the top to the bottom, where it's, okay, here's these two major groups of trees that have information that's relevant and both different from all the other trees in this tree set that we have. And so what's difficult about this is trees aren't just numbers, they're actually graphs. And so you can't go, I'm just going to take an average, like normal statistics. And there's also not like a known normal distribution of trees. And there's also not much agreement over how many parameters each tree has when you're trying to do like chi-square tests. And also, once you get a bunch of trees together, they exist in this multi-dimensional tree space. I'm still looking for a good graphic that has like trees floating in space. So if you know of any, let me know. And so what the current methods do is they use a lot of like tree to tree distances, which have their own set of problems. But they use multidimensional scaling or clustering algorithms to use the tree distances to kind of squish down all that data into 2 or 3D spaces, and then use different clustering algorithms to say, okay, identify this cluster, all of these are similar. And so the problem with that is that it's reducing this huge multidimensional tree space to 1 to 2 or 2 to 3 dimensions, and you're losing a lot of data and distorting things. And so since kind of my whole spiel is like, well, we want that variation, we want that information, we paid for the data, we might as well get everything we can out of it. So how do we identify trees without losing that information? So I got involved with a really great group of people working on Cloud Forest is the name of the program. It used to be called Treescaper. And they're basically using network community detection to group trees together. And so they're using networks. A quick overview of networks is you've got the nodes, which is where the information is. And then the edges tell you the relationships between the nodes. And so in this case, we've got one edge that's connecting the two non-mustache people. And this preserves information because you're not distilling it down to a 2 or 3D space. And so once you put your information in this network, you can do community detection. And so here, these guys will be a community without the poor mustache man. And there's a lot of research outside of phylogenetics that use these methods, because you can think of like social networks and that there's a lot of background looking into different algorithms for community detection and methods for networks. And so my part of this was looking at, can we actually use these networks to identify variation in tree sets? And so they'd done some preliminary proof of concept, but we didn't really know, like, is this actually going to work on empirical type problems? And so the other kind of cool thing about these networks is they use 2 types of information as nodes. One is trees. So each node would represent an actual phylogeny, and then the edges would be the tree-to-tree distances. But like I said, there can be some issues with the tree-to-tree distances and how they're calculated. They can be maximized by certain switches in topology that end up not being as helpful or meaningful as you want them to be. So we also implemented bipartitions as nodes. And here's just a quick, and I can go into this in much more detail, but I'm not going to here. But we also use bipartitions as nodes just to access the data differently and get different information out of it and get around kind of the tree distance problem. And so the nodes were bipartitions, and then the edges are how often those bipartitions are found in a topology together. And so what I did was simulate a bunch of difficult scenarios to say, okay, does this method actually work on phylogenetics? And so I simulated, some of the things I simulated were outliers, looking at, okay, if you have one tree that has really different information than all the rest, and also subtle differences in variation. So if you have two kind of signals going on in your data, but they're pretty similar, but still kind of have distinct, averages or information in general. And so I found out that it does work, which is great, obviously. And for the outliers, the networks where actual topologies were the nodes worked best. And then the bipartition networks were really good at picking up this kind of subtle structure. And so also we've got a paper in the works describing the bipartition network methods. And the empirical example that we used in that was looking at a big cat phylogeny. And so the network methods could actually identify areas of the genome on the X chromosome that actually had low recombination in those areas, which ended up agreeing with what the original paper from the paper we got the data from. And so that was really cool that without any information, you just kind of stuck in a bunch of scans of chunks of the genomes into the network. And it's like, yeah, we found exactly the same thing as the original paper. And so we can find really interesting biological patterns with this without previous knowledge of the system. So, all right, back. So that covers my trees and identifying kind of broad problems with network community detection. And in ways that lose less information and are potentially have a lot more expansion that we can do in different types of networks and community detection methods. There's a lot of research there that just hasn't been applied to phylogenetics yet, and tackle difficult problems with that. So next, I'm moving on to genes and morphology. I'm going to start with them lumped together a little bit. And so here, I was kind of asking a question, okay, if we know what the conflicting topologies are, we know what the conflicting signal is in our data, but we want to know how much support there is for one topology over another. How much of our data supports one or another? And so I used the wonderfully classic example of squamates because they have very fun diversity. And there's this historical conflict between the morphological and the genetic data. The morphological data and some of the early genetic data placed Iguania as sister. Oh, I've got a pointer. Iguania as sister to all other extant squamates. And that made a lot of sense in terms of ecology and morphology and behavior. But then when we started getting even just like multiple nuclear data sets, Iguania all of a sudden jumped way up into a group with anglomorphs and snakes that's collectively called Toxicofera. And so that really upset the squamate, some people in the squamate world, because the feeding and ecology of a lot of iguanids is not similar to anglomorphs and snakes. And so this seems like a good classic example where there are data sets published to look at how much data is actually supporting one topology over another. But since we generally know that molecular data is going to support Toxicofera as a group, I wanted to also look at the relationships within Toxicofera. I think some people say it Toxicofera. I say it Toxicofera. And so there's actually less consistent results in terms of the relationships within Toxicofera of the major groups across genetic studies. And so just to go over snakes, iguania, anglomorphs. I'm kind of cheating because I'm using a legless lizard, but they're cool, not a snake. And so the main hypotheses, the three different relationships that could happen here: iguania is sister to anguids, snakes and anguid sister, or iguania and snake sister. And most commonly in the literature, this is the resolution that you get. Some papers have high support for that with traditional support values. Some papers have medium support, but there wasn't much actually looking across multiple data sets to see what the amount of support was for any of these given relationships. And so that's what I wanted to do. And I also wanted to look at, as these bigger data sets are coming out, I also wanted to look at consistency across methods in which we're looking at support. And so I wanted to quantify support and also look at whether my quantification was going to change depending on what methods I was using. So phylogenetic support in big data, if anybody read the phylogenetic support issue of SysBio, you'd know, but I haven't read all of it, but I'll go over it for everyone else. So the traditional support values like posteriors, posterior probabilities, and bootstraps, they become maximized when you have these really big data sets, because it's looking at, based on your data, how sure are we that this is the topology? And it gets maximized. So you end up with different studies with maybe different analyses or different types of data, and you're getting completely different resolutions. And so one way to go about looking at the variation more carefully is on a locus by locus approach. And that's what's happening more frequently in papers. So basically comparing relative support for each topology that you're kind of thinking between. And so you're saying, okay, instead of just all of these loci support one tree, you're saying, okay, this set supports orange, another set supports red. And there's multiple ways to do this. The two that I looked at were Bayes factors and difference in gene log likelihoods. And so they're basically kind of hypothesis tests of, is this topology better than the other one, using likelihood ratios. And so Bayes factors are marginal likelihoods, and the difference in gene log likelihoods are maximum likelihoods. And the other thing that happens sometimes is people use these analyses or support values with thresholds, because you want to know, okay, is a support of 0.1 the same as a support of 8? So sometimes people will skim, okay, everything that has this amount of support and above, we're going to take those loci and do another analysis, and that's going to be our tree. So that's kind of why I was interested in looking across different methods if the numbers were consistent, because they are used to kind of harvest different parts of data, and we don't know how those compare across different studies. And so these are basically counting how many loci are supporting each topology. And so I wanted to know if the values are correlated. So to do this, I took a bunch of data of the squamate tree of different kinds, and I did pairwise comparisons of these different topologies, because you can only compare two against each other, and looked at the magnitude of support values and also how many were supporting one topology over another. And then for the methods, I also looked at the Bayes factors versus DGLS, which is the difference in gene log likelihood. And so what I wanted to see, here's just an example, is I wanted to see, okay, are these two correlated? If each of these dots is a locus or a gene, what we would hope to see is that there's a direct correlation between the two. If you get a 0 for one, you get a 0 for another. Versus if you're getting a 30 positive for one topology for Bayes factors and a negative 100 in likelihood, in maximum likelihood, then we've got a problem, obviously. And so what I found is that there actually wasn't strong support for any one topology in the Toxicofera relationships, which was a little bit surprising, since this is the most common in the literature. It did have some support. This is basically just counting the total number of loci that had positive values in support of one over another. And the majority, it doesn't show it here, but the majority of these values are actually pretty low support, which in some ways makes sense because the inner nodes in that part of the tree are really short. So there's just not a lot of information in terms of what the relationships are. And then in terms of the support values, I found that the low support values weren't as correlated as the higher support values. So here's all of them. And you can see that if you have all the data in here, we've got a decent correlation, and we've got that line that we're expecting of kind of a one-to-one relationship. But what I did was kind of remove the larger support values to see if it still held up or if those were driving the correlation. And so as I subsetted the data to say like the middle 60%, we really lost a lot of the correlation that we're seeing if you use all of the support values. So essentially the smaller support values are less correlated. And so do I have, okay, consistency across methods. So the low values have similar support for all the Toxicofera are low. And then the support values weren't as correlated when they were low. And I think part of this, sorry, I had something else on this. I think part of the reason is that for the maximum likelihood, we had a lot of values that ended up getting rounded to 0. And so there were a lot of just 0 values that I think was skewing things a little bit versus the base factors. Our guess is that the priors in the Bayesian framework were using the information that was there a little bit more. And so what you should do going forward depends a little bit on your opinion of priors. But in general, both of the analyses give us the same answer if you're just looking for broad strokes, like what are the loci that really support one topology over another. But if you're starting to count and using thresholds, it might be better to look at like the overall picture. And yeah, just be careful with the lower values and whether or not they're going to stay consistent across methods.
The dots there are individual genes.
Yes,
So there must be a lot of variation in how many SNPs or how much information.
Yes, yeah, there is a lot of information. And the further directions with this would be kind of figuring out, okay, what is it the information? What about these loci give them more or less support?
Can I ask you a question also while you're on
Yeah.
that page?
Mhm.
Just on the prior slide, can
Mhm.
you just explain really quickly what's going on in that lower left set of
Yes.
bar graphs? Because it seems like there's 3 columns for each of the 3 sets of.
Yeah, definitely. So since I could only do pairwise comparisons with these methods, this is comparing the purple and the blue topology across these 4 different data sets. This is directly comparing blue and green to each other, and so that there was more support for green, which is snakes and iguania's sister. And then this is comparing purple and green, and there is still some.
Represent in.
Oh, thank you. Yeah, so the third column is 0 numbers. So if you get a 0 value. Yeah. And this is just for the Bayesian analyses, but the maximum likelihood had very similar, just a lot higher zeros. Okay. So we've kind of figured out, right, low but similar support for different Toxicofera topologies if you're just looking at it agnostically, not assuming any biological sources and what's going on, which of course would be the next steps to this. And then methods, like I said, they're consistent for the most part, but not necessarily all the time. And so since I'm using the squamate example, the next obvious question is like, okay, well, what about the morphology? Why is it giving such a completely different tree? And also I was curious, is there any support in the morphology for the molecular tree? What we're guessing is the evolutionary history. And so also maybe we can learn more about morphological evolution. And so just to go over it again real quick, this is what the morphological data gives us for the most part. And so yeah, I wanted to basically like, okay, can we do what I just did with the genes, but with morphology? And so why I would have never said that I would ever have a project on morphology when I started, because I was like, doesn't matter, it doesn't mean anything. It's genes are the way to go. That's the answer to everything. Now I realize I was wrong. And I actually got interested in the models and the ways that we're making morphological phylogenetics and how it relates to our genetic methods. And it's also essential for a lot of things like the placement of fossils. We need to be able to estimate a morphological tree to be able to place fossils because you can't get DNA out of everything. And you can also learn about morphological evolution, which in Molly Womack's lab, I'm slowly being convinced that that's also cool. And so what we would want to do is group traits with similar evolutionary histories, right? Because everything with the genes, it's based on loci. So you need kind of the set of information that evolves similarly to be able to use the Bayesian models and more complex models other than just shoving it all together and running it through maximum likelihood, which is possible and gives you a perfectly fine tree, but we don't learn a lot about the variation in the information. And we can't use as complex of models. So the problem is really what's the morphological equivalent of genes? And so I came across this idea of in the literature of modules of evolution of like, okay, these groups of characters might be evolving together. But for a lot of things, this is completely unknown, right? Like squamates, no, not sure. And so then it was like, okay, so can we quantify, support, and identify things that are evolving together, right? Because if we're looking at what's supporting one topology over another, we're also learning that those are all evolving in a more similar way. So we want to group traits together. I want to do it in a Bayesian framework because I'm biased. And so what I came across trying was using a mixture model. And I'll go over that really quickly. Basically, a mixture model is it has a parameter that you're estimating. You can give it classes, kind of like think of them as bins, and they're subsets of the model. And so what you're doing is you're both estimating parameter values, and you're also looking at the preference of each character in your data set to one of the classes or one of the bins. And so one model or one example that's used in genetics is the cat model, if anybody's heard of that, which is not the same as like Raxmel cat. And so what it does is it basically has bins for different equilibrium frequencies. I think it's used most often in like proteins, protein alignments. And so it looks at different equilibrium frequencies of proteins throughout the tree and models that. So you can say, okay, this set of characters is more likely to have evolved under this model that's different than this other model, rather than making everything conform and estimating the exact same parameters across the entire alignment of data. And so, okay, what if we do that with topologies? And then essentially each class will be a different topology or topological constraint. And then we can identify which characters, you know, are more likely to support one topology over another. And so this has been used in genetics to identify recombination breakpoints in like genome-wide data. And also, if anybody's familiar with Bucky, clustering genes by topology, it also uses a mixture model. And so what's cool with this is that we can actually both estimate the tree and assign characters to a class at the same time, rather than first having to estimate the tree and then look at the support for the characters. And so, like I said, each class is a topology. We're estimating the characters for each class separately. And to figure out, you know, which class a character prefers or is assigned to, we're basically looking at, during the analysis, how much time is it spending in one class versus another. Like I said, it's in the Bayesian framework. And so when I wanted to do this, there wasn't actually any software that would let me do it. So I talked to the RevBase people and they helped me figure out how to edit some of the code to actually fit in both topologies as classes in a mixture model and then also shove morphological data in it. And so the data that I used for this is from Samoy's 2018. Really awesome, huge morphological data set for squamates. And so I'm just going to go over really quick interpreting character assignments to different classes, what we might expect. And so if the characters have no preference for one topology over another, you know, they're saying based on my information, either one of these could be true. You'd expect to see a graph that looks like this. And so on this axis, we have number of characters, and this is time spent in each topological category. And so this is one topological category, and this is another. And based on where the bars are along here, if they're over here, it'll support this one more. If they're over here, it'll support this one more. So this would be an example of the majority of characters are spending 50% of their time in one and 50% of their time in another. So there's not really information that says, okay, yeah, this is more likely topology. So I colored in blue. Another option is that there'd be a strong preference and the majority of characters agree. So for example, it would be majority of characters prefer the purple phylogeny and support that one, and maybe just one or two spend most of their time in the other topology. And then there could also be strong preference or conflict. So having, you know, half of the characters are more like, or have signal that matches this topology, and the other half have information that matches the other topology. And so what I wanted to apply this to the squamate data to see, okay, are there certain characters that are maybe overwhelming the signal to give us the Iguania as sister to all other squamates topology? Is there a couple that have really strong signal? Or are there also characters that will support the molecular topology more? And so what I found is that kind of what you would expect, which was neat, is that the majority of characters, the slight majority of characters, have signal that supports the Iguania as sister to all other squamates. But there are still some that support the molecular Toxicofera hypothesis. And so this is the example that I showed of there's strong preference and also conflict. And so my next question was, okay, well, what parts of the body? You know, is it the hands that are all supporting one topology over another? And so this also gets to kind of the modules of evolution. And so I looked, okay, I wanted to ask, are there anatomical modules in squamates? Do subsets prefer the same topology? And so I split it up into anatomical subsets just based on characters that were close together anatomically, because I'm still not a morphologist, despite the fact that I am working on some morphology. And so I wanted to see, there we go. Say for limbs, do all the limbs prefer one topology, or do some of them prefer one and some of them the other? And so essentially what I found is that there weren't really any of these large clusters that I was grouping them into. There was not, they did not prefer one over the other. They were mostly split, except for dentition. That was the one exception. And I think partially this is because it's just a proof of concept. Like I said, I'm not a morphologist. The modules that I predicted were pretty broad. And also this data set was specifically developed to not have correlated characters. So that also is probably why I didn't find a whole lot of correlated characters morphologically close to each other, which is cool too. But I really want to work on this more and do more simulation testing and explore the opportunities for this model, because we can get at some cool evolutionary questions. We can't, oh boy, okay. Let's see. Let me see if I can get the, nope, we're skipping that slide. Okay. Like I said, it's been a while since I made a PowerPoint. So to summarize kind of the phylogenetics part, what I get really excited about is all of the information and variation that's in these giant data sets that we collect. And we can learn really cool things from using these methods to kind of better quantify the variation and understand it. So in trees, I was looking at the more broad patterns, working with kind of these multidimensional, mathematically difficult tree spaces, and using networks and community detection. Making these cool little network models. And with genes, we're looking at quantifying support once we kind of already know what the conflicts are. Let's see how much time. Ooh, okay, we're speeding through some of this, sorry. And then morphology using mixture models to kind of quantify support and test modularity. And so on to frogs, which are just awesome. So for my NSF postdoc, I proposed looking at evolution of trait loss. And then I also got involved in some side projects in Utah with conservation genetics. And so I will not be featuring vocal sacs. And so how did I get back to animals from there? Like I mentioned, I was really originally in a more empirical lab. And as I was working and collecting these giant data sets, I really wanted to know how the models interacted with this huge variation in data. And if the lab work has to change for different organisms, why aren't we changing the methods and models as well? And so that's where my agnostic approach to a lot of this came from. And so I really wanted to return back to more empirical work after doing a like super deep dive into methods. Oh, good. Messed up some more of my slides. Okay. It says stuff, I swear. So yeah, I wanted to kind of experience and do some research on the other side of the phylogenetics. Once we do have a single tree, somebody else did all of this other method stuff for me. I have a single tree. How do I use it? Okay, there we go. Yes. So the first project is the trilgon, loss of vocal sacs in anurines. And so it's interesting that they're losing vocal sacs because it seems to be a sexually selected trait, right? They're using it to attract mates. And then I'm also working on a conservation genetics project. And these are both inspired by boreal toads or western toads or subspecies of California toads here as well. And they don't have vocal sacs. They're kind of the species complex, and some of the species that are closely related do have vocal sacs. And so it's just this really interesting system. And then they're also at risk in Utah. So I got involved working on some conservation genetics with that. So vocal sacs enable loud calls. Is it good? Right, there's my Gulf Coast toad from Louisiana showing off his impressive vocal sac and calling for the ladies. And so vocal sacs are a secondary sexual sex characteristic. They're only in males, except for like one exception, maybe in one species that I couldn't name at the moment. And they're using it to attract females and defend territory. And so it's really interesting, like if this is a sexually selected trait, why is it lost? Where is it lost? How? What is going on? And so some other common sexually selected traits, big horns, antlers, flashy colors in birds, and then we get to add the toads to the mix. And they're often really big and showy. And so a lot of research focuses on gaining these traits rather than losing them. And there aren't very many studies across large clades looking at potential mechanisms of why they're lost, or even just patterns of loss of sexually selected characters. And so I'm looking at vocal sacs across anurans, trying to figure out kind of the who, where, and when of this loss. And so to look at the who, I'm looking at presence, absence of vocal sacs across species, where ancestral state reconstruction to figure out where it's been lost. And then the why is starting to identify potentially correlated characters to get at hypotheses of why this might be happening. And so for the why, a couple of the hypotheses I'm looking at are breakdown and signal transmission. And for that, I'm looking at correlated characters of habitat, whether there's loud habitats like torrential mountain streams. I'm also looking at loss of ears. If you don't have ears, are you less likely to have a vocal sac because you're just not using that sense as much? And then I'm also looking at potentially an overall change in female preference, looking at whether or not multiple secondary sex characteristics are lost at once. I'm using nuptial pads as my other secondary sex characteristic. So I got this NSF grant literally like the day before campus shut down in 2020. So my original plan was to focus on bufonids and examine museum collections, which I would have been here, but it seemed like not a great time to move here and hang out. So what I've ended up doing is a literature search across all anurans, and it's ended up being pretty cool because I've been able to include 10 undergraduate researchers so far by just having them do lit searches over Zoom, basically. And I've had people from 3 different campuses, including one from here, who just started recently. And so I've been able to actually provide research experience at a time where not a lot's going on. So here's the total number of recognized anuran species, genera, families. I have about 1,500 species with vocal sac data currently. Of those, about 17% of the species lack data. About 16% of the genera are variable, meaning they both do or don't. They have species that do and don't have vocal sacs, and about 35% of the families are variable. So it's definitely a trait that's not being lost, you know, in larger clades. It's not like, okay, this large clade, none of them have vocal sacs. That happens some, but there's also a lot of just like individual or smaller clade instances of loss. So I'm looking at that. I'm currently in the data collection. Next, I'm putting it on trees, which I thought I'd have for this, but I'm not used to that kind of analysis. So still working out the kinks. And then quickly, the conservation genetics. I'm working on the boreal toads in Utah, and there's a lot of isolated populations that are declining. And so I got a grant to work on the genetics in Utah to figure out kind of captive breeding programs and where if they can introduce them from other populations. And so the boreal toads are this whole, they're all over the place. But there's a lot of kind of isolated populations, and some of them have been described as new species. So the people in Utah are going, okay, well, do we have a new species? Should this be an endangered species? And so yeah, I got funding through the Endangered Species Mitigation Fund, and I'm working with the Utah wildlife people on this project. And again, here, these are the populations that they're kind of wondering, okay, is that different? They're really isolated from the rest. And which populations are more closely related. And so I've actually got to go collecting a little bit, which is fun. And I collected some specimens, which I'll deposit here, which will be some of the first outside of California boreal toad specimens with genetics, because all of the sampling in museums is from California, which made this a little bit difficult at first. And I also got to go to the breeding program and swab toads in the mouth to get genetics, which was kind of fun. So I'm actually getting to go out and then sequencing it. And so I will go over this quick overview since I'm running out of time. Apologies. Yeah, we kind of looking at variation in phylogenetics is one part of my research. And then the other part is I'm starting to get back into more empirical stuff, and I wanted to get back into working with state agencies because it really helps inform the methods part of my research. And with that, thanks to LSU and my advisors for the help with the phylogenetics and frogs, to Becca and Molly and labs and the UDWR people, NSF. And with that, I'll take questions.
James.
Far in the back. Yeah, you.
Oh, yeah. Awesome talk.
Thank you.
Did you apply any of the mixed models to any of the traditional morphological data sets, like in the JOCs or any of those other ones?
Good question. For the people on Zoom, the question was, did I apply any of the mixed models to the Gauthier older data sets? I don't think I did. Why? I don't remember, but it's definitely something that I will do in the future. Yeah, don't remember why I didn't. Good question. So I don't know. Yeah.
So do you have an assessment though of that sort of controversy between Toxicofera and the traditional morphology analysis you undertook? For one of the things I was thinking about was whether you actually took those characters that were sorted into the alternatives and just analyzed those to see what they had to say about the overall phylogeny, because it's more than just this one question, right? The whole tree is kind of structured.
Yeah. Yeah. So repeat for the people on Zoom. Kind of what from the morphological kind of subsets that I did find of the ones supporting one tree over another, what happens if you just take those characters and analyze them and make them their own tree? I have not done that, but that would definitely be a next step to figure out. And yeah, to see if it would actually come out with that tree. Yeah, that project was kind of a proof of concept, and I'm really excited to like do all these next steps at some point. So yeah. Thanks. Yeah.
So when internodes are close, you expect incongruent phylogenies, and with the scale over which you expect those incongruencies varies, I mean, among species across the genome, sort of depending on things like recombination rate variation. Can the approach you described be used to identify the scale at which to look at genomic segments? I mean, you mentioned this phylogeny of cats where the X chromosome stood out. Can you use your approach to ask, should I be looking at 100 kb chunks or 1 megabase chunks? Or
Okay, and the mixed model or the locus by locus?
Locus by locus.
Locus by locus.
But I'm thinking more in the case where you would have whole genome data.
Yeah. Okay, so let me repeat the question. You can tell me if it was what you were asking. Is basically, can you use the locus by locus support values and compare different size chunks of the genome to figure out what size chunks to be using as individual loci?
Yeah.
Is that what you're?
Yeah, but not so much in data sets where you have, say, 100 loci, but in data sets where you have full genome sequences, and you can make the decision,
right?
What size chunks to chop the genome into.
Yeah. I mean, I would assume so that you could. Are you thinking like comparing the different sizes that you've chopped them into to see if those have more or less support for one topology, or? Well,
I'm just thinking that you expect congruence among regions that have similar evolutionary histories. And the size of that region that has a common evolutionary history is going to be dependent on things like recombination rate. So if you're in a low recombination region, that's going to be bigger, right? And so ideally, you'd like to discover biologically the regions of the genome that provide support for one over another.
Yeah, definitely. I think that there is a program that does something similar to that called Saguaro. If it's not Saguaro, it's a different cactus name. But it does similar to what you're talking about, is it identifies regions of the genome that have similar topologies. And I used to know a lot more about the program. I don't remember. It might also use a mixture model of the different potential topologies, and then it looks at which sections of the genome fit into which topology. Does that answer it?
Yeah.
Close enough? Okay.
Great talk. I'm curious about the details of like the information that you were looking at in the literature.
In the which part?
Literature for characterizing vocal sac loss.
Oh, okay, yeah, literature, got it.
So I was wondering if you explicitly look for loss of vocal sac or traits that might be correlated with calls like vocal beats or
Yeah, so that's a good question. That I've answered a lot from my undergrads. So yeah, I'm looking at explicitly papers that say there are no vocal sacs. And one tricky thing is sometimes it says there are no external vocal sacs, and that doesn't count because some things are described as internal vocal sacs, and there's really interesting literature that's come out recently in terms of like the different types of vocal sac morphology that you can have across species, which is like a whole nother thing that's really interesting. But yeah, I'm mostly looking at whether it says no vocal sac or no vocal slits, and I am also using, if they do have vocal slits, I'm assuming that they have a vocal sac, and if it says that they don't, I assume that they don't have a vocal sac.
I was just asking because I found that usually vocal beats are difficult to characterize, and sometimes you can say no vocal beats in the description, but they usually do have, or sometimes it happens that they do have vocal beats.
Yeah, and I think to that, oh, sorry, I didn't repeat the question for people on Zoom. The question was basically how I'm characterizing vocalization, vocal sacs in the loss of vocal sac research. And one thing that I want to do is look at actual vocalization versus presence or absence of vocal sacs. So I think what you're saying is there are some frogs that will vocalize despite that they don't have vocal sacs. It's just much quieter. So I'm kind of going with the course, like, are they really loud and using this to attract mates versus smaller?
I think both. I think also sometimes it's just difficult to look at preserve the specimen.
Yeah.
And like correctly, like make a right characterization of the vocality.
Yeah.
So in both cases.
Yeah. No, I definitely agree. And I haven't spent that time yet with specimens. I'm so far just doing the literature, but I'll come talk to you when I do. Any other questions? Any questions on?
Oh,
Oh, no, I was just gonna say any questions on Zoom, but I don't see anything, so. Speak now. Nope.
Great, thank you very much.